Hello, welcome to my lecture video which entitled is Chloroplast Genetic Markers. Before I proceed to this lecture video, I will introduce my name first. My name is Valerie Heron from Conservation Biology. The first content that I will talk about is evolutionary history for chloroplast. As we can see in this picture, it says that the evolutionary history of chloroplast means that mitochondria and chloroplasts likely evolved from engulfed prokaryotes that once lived as independent organisms. At some point, a eukaryotic cell engulfed an aerobic prokaryote, which then formed an endosymbiotic relationship with the host eukaryote, gradually developing into a mitochondrion. Eukaryotic cells containing mitochondria then engulfed photosynthetic prokaryotes, which evolved to become specialized chloroplast organelles. Also, as we can see in the picture, there are two steps, which is primary endosymbiosis and secondary endosymbiosis, which means primary endosymbiosis is before the cyanobacterium engulf to the eukaryotes. The secondary endosymbiosis is after the the cyanobacterium, photosynthetic bacterium already engulfed the eukaryotes. Next is about function and structure of chloroplasts. First, I would like to talk about the altar structure, which is the three main components of chloroplasts, which are classified as envelope, stroma, and thylakoids. First, envelope. Envelope is the entire chloroplast, which is bounded by an envelope, which is made of a double unit membranes. Across this double membrane envelope, exchange of molecules between chloroplast and cytosol occurs. Second is stroma. Stroma is the matrix of stroma fills most of the volume of the chloroplast and is a kind of gel fluid phase that surrounds the thylakoids, which is the name is grana. It contains proteins, ribosomes, and DNA. The stroma is the site of CO2 fixation and where the synthesis of sugar, starch, fatty acids, and some protein occurs. Third is thylakoids. The thylakoids consist of flattened and closed vesicles arranged as a membrane network. Thylakoids may be stacked like a neat pile of coins, forming grana. There may be a 40 until 60 grana in the matrix of a chloroplast. Okay, in this component, which light reaction occurs in thylakoids membrane. The function of chloroplast is photosynthesis. There are two steps of the reaction. The reaction, which is the light reaction and dark reaction. Light reaction is the photochemical reaction, where also known as Hill reaction, where photosynthetic electron transfer reaction. In light reaction, solar energy is trapped in the form of chemical energy of ATP and NADPH. During it, oxygen is evolved by photolysis. Second is dark reaction, also known as Kelvin reaction, where photosynthetic carbon reduction cycle, or also known as PCR cycle, or carbon fission reaction or thermochemical reaction. The reducing capacity of NADPH and energy of ATP is utilized for the conversion of carbon dioxide to carbohydrate. This dark reaction occurs in the stroma. Next is gene with their function in the chloroplast. As we know, chloroplasts have the similar genomes to those of mitochondria in that they consist of circular DNA molecules present in multiple copies per organelle. Chloroplast genomes are larger and more complex than those of mitochondria, ranging from 120 to 160 kb and containing approximately 120 genes. First genes in the function in the chloroplast is the RNAs, which is function as translate all the mRNA codons according to the universal genetic code. This organelle genome is encoded both the ribosomal and transfer RNAs used for translation of chloroplast mRNAs. These include 4R RNAs, which is 223S, 16S, 5S, and 4.5S, and 30 tRNA species. This gene is one of the most commonly used in function of chloroplast, which consists 30 tRNA species for genetic apparatus function. This gene is sufficient to translate all the mRNAs codon according to the universal genetic code. Second is ribosomal proteins. Ribosomal proteins function as regulating translation of chloroplast mRNAs, including light-regulated translations. So, addition to these RNA components of the translation system, the chloroplast genome encodes about 20 ribosomal proteins that represent approximately a third of the proteins of chloroplast ribosomes. This gene is the second commonly used for genetic apparatus function in chloroplast. The third gene is AT2G2128T, or known as GC1, which is giant chloroplast 1, mostly composed of a conserved domain of NADP dependent epimerase. Okay, which use nucleotide sugar substrates for a variety of chemical reactions. The sugar epimerase is widely found in animals, plants, and microorganisms. Initially isolated from E. coli, catalyzing an epimerization reaction through the transient reduction of NAD+. Once an epimerase gene of bacteria is mutated, it can cause reduced infection, decreased pathogenicity, and high sensitivity to antibiotics. So, based on the facts of the giant chloroplast 1, it modify and regulate the carbohydrates attached to the surface of the membrane of chloroplast inner envelope in the stromal site. And this gene is one of the rarely used function in the chloroplast. The fourth gene that rarely used function in the chloroplast is PSBA. PSBA is an indispensable component of oxygenic photosynthesis, which has two types of function, which is molecular function and oxidoreductase. For molecular function, it consists of biological process and ligand. For oxidoreductase, is electron transport, herbicide resistance, photosynthesis, transport, and the component are calcium, chlorophyll, chromophore, iron, magnesium, manganese, and metal bind. This is the gene of protein coding genes and non-protein coding genes. Okay, why are the two genes more commonly used than the other two genes? This is because tRNAs and ribosomal protein, this gene is one of the important function in chloroplast, which is translating and regulating the mRNAs codons according to the universal genetic code. Compared to rarely used gene, which is giant chloroplast 1 and PSBA, because giant chloroplast 1 is since the main results of the previous studies that AT2G1280 is an important chloroplast division gene cannot be confirmed by our intensive study, and large chloroplasts are rarely found in the mutants. For the second gene is PSBA, which induced mutation are simple deletion that easily eliminate the PSBA gene, and this clearly give the explanation why the two genes are commonly used gene compared to the other two genes.